So now we're going to move on to the lightning talks, our first round of lightning talks, which is still related to fundamentals in haptic perception. Lightning talks are brief 8-minute talks with 2 additional minutes for questions. And our first presenter will be Dr. Gordon Legge. Dr. Gordon Legge is a distinguished McKnight University professor in the Department of Psychology at the University of Minnesota, and he's the Director of Graduate Studies in the department. Gordon is also the head of the Minnesota Laboratory for Low Vision Research and is the founding member of scientific co-director of the Center for Applied and Translational Sensory Science. So please join me in welcoming Dr. Gordon Legge.
Okay, thank you. So unlike Nick, I do have slides. And unlike Nick, I have a tactile demo that's being passed around. It's a tactile acuity chart, which I will refer to. But hopefully you can pass it along and kind of feel it. So my talk is called Tactile Acuity in Blind and Sighted Subjects, the Impact of Life Experience. Before I start, I just want to mention that I didn't know Val Morash well, but enough to really appreciate her talent and creativity. Actually, we tried to hire her as a postdoc at Minnesota. We failed because she had such a great situation here at Smith-Kettlewell. And we're certainly going to miss her. She was a great colleague and a great contributor to a very important field. So could we go to my first slide on 2 studies. So today I'm going to briefly try to rip through a description of 2 studies, one published in 2008 on tactile acuity across the lifespan in sighted and blind subjects, and then a more recent follow-up on pianists. So our work built on 2 previous findings, and they should be summarized in the next slide. First, blind subjects have better tactile acuity than sighted subjects. Blind subjects can resolve gaps between 2 dots that are roughly 15% smaller than sighted subjects. Secondly, both blind and sighted subjects display an age-related decline in tactile acuity. Threshold gap size on the fingertip increases about 1% per year, and from age 20 to 80, that results in a doubling of the threshold. So this age-related decline should make Braille reading more difficult in old age. But it seems that people who learn Braille early in life continue to read Braille fine into old age, and I'm a living example of that. So we were curious about this apparent contradiction. So we decided to re-examine the age dependence of tactile acuity. And we have the next slide, which shows 2 charts. So one of the 2 charts I'm going to describe here is an example is being passed around, the Landolt-C chart. So we designed 2 styles of tactile acuity charts sort of based on similar principles to eye charts. The dot chart, which is shown on the right, has symbols similar to Braille. So subjects actively move their fingers across the symbols, and this kind of active scanning distinguishes our testing from most earlier studies of tactile acuity, which use passive stimulation of the fingertip. The 4 test symbols on the dot chart are essentially the Braille characters D, F, H, and J. And all of these have 3 dots, and the symbols differ only in the location of the missing 4th dot in the square of dots. So when Braille readers do this chart, they just read off the letters. When sighted people do this chart, they indicate the missing dot, for example, upper right. The chart has 9 lines, 8 characters per line, and the dot separations decrease by 0.1 log unit per line. That's about a 26% reduction in size. The dot spacing on the 6th line of this chart, which is labeled the 0 line, corresponds to standard Braille character spacing. And we score this acuity in the same way that eye charts are scored, so-called Logmar scoring principles from eye chart design. Our second chart, which is shown on the left, uses the embossed Landolt C rings, Cs with a gap. And this time the four symbols are basically just defined by the orientation of the gap in the ring. This is the one that's being passed around. The ring, these C symbols are kind of low in linguistic meaning and roughly equal salience for blind and sighted subjects. And the chart I'm passing around was based on the design we had, but it's a new version built by Steve Landau of Touch Graphics. I don't know, is Steve here? Anyway, it's...
The flight was delayed. He's going to be here around noon.
Okay, great. Anyway, it's a nice design of the C chart. Could we go to the next slide? So in one of the experiments, experiment 2 in our 2008 study, groups of 22 blind and 19 sighted subjects were split into young and old groups, and they were tested with both of these, the C chart and the dot chart. And the next slide. So this slide shows tactile acuity data versus age. The dot chart is in data on the right panel, and the ring chart or the C chart in the left panel. Blind subjects are the red symbols. Sighted subjects are the blue symbols. The vertical scale here shows threshold spacing at the acuity limit. It's a logarithmic scale on the left and millimeter scale on the right. So larger values mean poor acuity, need bigger gaps. And the pattern of results is the same for both charts. So consistent with the previous findings, the rising regression lines for the sighted subjects represent the decline of tactile acuity with age. And also consistent with previous findings, tactile acuity is better for the blind subjects, the red symbols, than for the sighted subjects, the blue symbols. But the novel finding here is that blind subjects exhibited no decrease in acuity with age. The red curves are flat. In other words, the blind subjects retain high tactile acuity across the lifespan in contrast to the age-related decline in tactile acuity for the sighted subjects. So why this difference? Well, it's possible that a lifetime of intensive tactile experience might protect against age-related decline of tactile acuity for the blind subjects. If so, we asked if sighted subjects who regularly devote more attention to tactile function might also exhibit preserved tactile acuity into old age. So we decided to test proficient pianists given their attention to fingering and dynamics. We go to the next slide. So we recruited and tested groups of young pianists, mostly piano majors in our School of Music at the University of Minnesota, and older pianists, mostly piano teachers and advanced amateur performers. These were all normally sighted subjects. In the next slide. So here are average data comparing performance for the young and old pianists and the sighted and blind groups from that earlier 2008 study. And if you look first at the data for the ring chart on the left, the sighted pianists, the green symbols, are very similar to the blind subjects, the red symbols. And they show no effect, basically no effect of age. In other words, sighted pianists appear to retain high tactile acuity into old age. For the dot chart on the right, the pianists lie between the sighted and the blind group. So perhaps the blind subjects have some added advantage on the dot chart in distinguishing the Braille symbols. And the next slide. So we conclude from this that lifelong experience with focused attention to touch may act to preserve tactile acuity into old age for both blind and sighted subjects. Regarding Braille reading, barring health problems affecting the fingers, blind readers retain adequate tactile acuity for proficient Braille reading into old age. But the age-related decline in tactile acuity means that many sighted subjects, most sighted subjects, maybe not pianists, lose tactile acuity and would likely run into trouble learning Braille in later life. In the last slide, just a list of collaborators on the 2008 and more recent project, and credits for funding to, thanks to NIH and the Helen Keller Foundation. So thank you.
Great, thank you so much, Gordon. So if anyone has any questions, they can raise their hand and Daisy will bring you the mic. And just please say your name before you start talking. So we have time for like 2 questions. There's one question in the back.
This is Ken Perry with the American Printing House. I started learning to read Braille at 21, and up until recently, I'm just now starting to read. Well, I'm almost done with my first book on the Order Reader 20 because it's a much harder Braille. Did you find that? I mean, if a person hadn't learned till later, it's harder, even though they're still young. Because I mean, I was 21, and the Braille paper is too squishy for me, and the older Braille displays were too squishy, but this new one is harder. So I'm just wondering, is there something when you're dealing with younger readers that have never done tactile reading type stuff?
Yeah, that's an interesting question. And actually the data we actually had data from something like 44 Braille reading subjects. And it was hard to find much correlation between their acuity and history of or experience of Braille reading. We might have had some floor effects that made those effects harder. I think there have been a more recent study by Wong and Goldreich. I think they did find some relationship between some measures of tactile acuity and age of onset for learning Braille, number of years reading Braille. So yes, I think probably our effects are a little hard to find, but I think they're there.
Gordon, I would like to point something out that occurred to me because of my own involvement in visual attention research for a number of years. When we talk about attention, and you mentioned the word also, we sometimes think about 2 very different tools, if you want to call it that. One is spatial in the sense, like pay attention to something here or there. But you can also pay attention in parallel to everything that's green in a picture that you're looking at. And I think it's interesting that you mentioned attention, because paying attention to the interpretation of, let's say, a dot pattern is very different than spatial attention. It's more an interpretive kind of attention. And I think that needs to be taken care of, needs to be taken into account when we try to understand why Europeanists, for instance, do something that the rest of us usually don't.
Yeah, I don't know quite how to respond to that, except to say that I think blind and sighted people are touching things all the time, like coins, for example. But I think sighted people are relying more on visual attention to distinguish the coins. Blind people may be relying more on touch. So it's kind of a coarse sense of what I mean by attention. And maybe your thinking about it would be helpful as a more refined way of thinking about attention.
Gordon, do you have any thoughts about older people who might normally be expected to have lost acuity training up?
I think there is some evidence that training can help enhance tactile acuity. I'd like to know, I keep asking, are there examples of late blind individuals who learn to become proficient Braille readers? And I've never really had a straight answer about the success of that kind. Yeah. Is there? Great, good.
So like I said, I went blind at 21, well, 20 going on 21. I think it has to do with, boy, I was going to say GS, but I'll say giving a care or caring, because I've never been really interested in getting really good at Braille because there's such good audible interfaces. But now that I have a tactile display, I'm getting really good at tactile graphics, but I'm not excellent at Braille. So that tells me I still have really good touch, but I don't care to learn Braille. Does that, it sounds awful, but when I have such easy access to computers and audible, because I learned it, I lost my sight in 1991, and, like ASAP and JAWS, they just started coming on strong, and I've never really had to, I did my calculus with an editor, or in my mind, because, I could do like a 5-page calculus problem just in my mind with audibly. So Braille to me was never that important, although now I'm learning, if I did learn Braille earlier, things would have been so much easier. So it's getting better now. So there you go. That's my 2 cents nowadays. And I think that's what's happening to kids. They don't care. They just, so there you go.
Okay, so I think that's it for this lightning talk. So can we all thank Gordon once more? Okay, our next lightning talk will be given by Dr. Lora Likova. Lora is currently a lab director at Smith-Kettlewell here. Her goal is to integrate multiple brain imaging techniques, including MRI, fMRI, coupled with a unique motion capture system, diffusion tensor imaging, tensor-based brain morphometry, and EEG to study neuroplasticity, brain mechanisms, and art and learning, rehabilitation of blindness, memory and spatial cognition, vision deficits and TBI, and multimodal sensory motor processing across levels of visual impairment. So now Lora will be presenting her talk, her lightning talk, 8 minutes and 2 minutes for questions, the cognitive-kinesthetic rehabilitation training and brain plasticity and blindness. So let's welcome once more Lora Likova.
Hey, thank you. So in 8 minutes, I'll try to cover these 3 points. To tell you something about my cognitive kinesthetic rehabilitation training, which is based on memory drawing, how it drives brain plasticity, and how it generalizes its effects beyond the trained modality. So this is a method I developed a number of years ago. The idea was, so as you know, in blindness, the eye-hand coordination mechanism is lost. So the idea was to develop a method that will help to enhance the memory representation, so that, figuratively saying, to replace the eye-hand by memory hand or mental map hand coordination, and using the haptic modality as the input. I can't elaborate and describe the training, but I'll give you a general idea. So as a training task, I chose drawing from memory. What is shown on the screen are a few examples of the raised line battery. You see objects and faces, a portrait of George Washington, a vase with a flower, a boot. These are a few examples. In other words, the participants had to directly, so to say, jump in the deep water. We don't start with lines and simple geometric figures. The whole training takes only about 10 hours. This is 5 sessions of 2 hours a day, preferentially on the sequential days. So what is shown here are pre-training drawings. You see some formless figures, and post-training, you see well-recognizable images, well-connected, well-coordinated. By the end, people learn to recognize and to draw in less than 20 seconds, which is necessary for the fMRI part. Here I have a movie, and I have to thank Christio Mine for making this movie. In the upper left corner, we see a famous video of Picasso drawing a vase with flowers, and the bottom right is one of our blind subjects. She's additionally blindfolded here for the video. She's exploring a raised line image of this vase with the flower, notice with left hand, and now she's drawing freehand from memory with the opposite hand, so no simple muscle memory. And pay attention how fluently, how confidently she is drawing. I would say, competing with Picasso, at least in this respect, yeah. So this kind of post-training results are telling us at least two things that the training is achieving. So this is another video, simply as a background of drawing the Washington. It's telling us that people really learn to develop rapidly precise and robust internal representations, and moreover, learning to use them to guide precise motor activity. So drawing this image requires a lot of, you see, you have curves, you have straight lines, it's quite complicated. And I have to tell you that these sight people cannot do it visually so well from first time. So the second point is generalization or transfer of training hypothesis that I had based on the nature of the training. So this cognitive kinesthetic training operates in a way that it simultaneously orchestrates multiple perceptual, cognitive, and motor components. So engages the whole perception cognition loop. People usually talk about perception action, but it's perception cognition action, and my emphasis is on cognition. It involves a whole list. I don't have time to read it, spatial memory, spatial analysis, and so on. And supporting this hypothesis, we become friends with people because we are for a number of days, they come again months later. They were reporting improvements in other areas of their life. So we decided to test this hypothesis, running before and after the training, standardized cognitive test for the blind, which I found through the American Federation for the Blind. And I'd like to make a brief point that this is a whole other big field in haptics. There is a whole, I could say, industry for spatial tests for the sighted. It was a huge problem to find standardized tests for the blind. It's blind and low vision. It takes about 2 hours. It has very high test-retest reliability and measures a whole range of core spatio-cognitive abilities. So results showing that our hypothesis was confirmed. So what you see here, blue and red bar is pre to post training. So you see highly, significantly improvement on the cognitive test for the blind. So although the test has well-established very high test-retest reliability to be super safe, we also ran a control group, and you see that without training, there is no improvement. The question was how long the improvements are maintained. So we call people again 3 to 6 months later. Some people up to a year later, actually, but this is the green bar. You see that there is no significant reduction in improvement. In other words, the improvements are maintained at least on a time scale of many months, which implies that the cognitive kinesthetic training can serve as a wide spectrum rehabilitation through a single and quite a fun task. Okay, so we see rapid, dramatic behavioral changes, but what I am especially interested is the brain mechanisms underlying the changes. So to study brain plasticity driven through this method, we ran also functional magnetic resonance imaging before and after training. It's with different populations of blindness, whole brain, at several time points and so on, and the drawing engages widely distributed brain network. So there are a lot of interesting findings here. I'll stop very briefly on one of the earliest findings because it's most related to the mental maps we are talking here. So these studies in the blind led to the finding that the so-called in working memory visual spatial sketch path is implemented in the primary visual cortex V1. And because my findings were covering, also congenitally blind, I proposed that this is not visual spatial, but a model spatial sketch path, which makes a lot of sense. Only to come to the previous discussion, the best hardware for precise spatial kind of projection screen. So the idea of this sketch path, you somewhere project the mental images, inspect and manipulate them, is the primary visual cortex. It is the largest topographic map in the brain with the highest spatial resolution and parallel processing. So recently, very briefly, we assessed again this proposal using Granger causality. So if this is a memory sketch path, a model memory sketch path, it has to communicate to be influenced from high order memory structures, and the first suspect, of course, was the hippocampus. What you see here is not activation, causal influences, blue to red, pre to post. You see the hippocampus, the blue bars, is not interested in V1 before training, and then you have these beaming red bars showing significant improvement memory influences from hippocampus to V1, and similarly another hippocampal complex area, the same pattern. So in summary, the cognitive-kinesthetic training drives brain plasticity in a top-down manner from high-order memory structures to V1 and enhances memory, spatial motor cognition, but spatial motor coordination, but beyond that, it transfers or generalizes to untrained cognitive abilities. And I'd like to thank my collaborators, Christian Spiro, Laura Christopher, and my NIH and NSF grants. Thank you very much.
Okay, thank you so much, Laura. Unfortunately, we don't have any additional time for questions, but during lunch or during one of the coffee breaks, please feel free to ask questions during those times as well. Our next presenter will be Dr. Santani Teng. Santani is a postdoctoral researcher here at Smith-Kettlewell. And he's a research affiliate in the Computational Perception and Cognition Group at the Massachusetts Institute for Technology, MIT. Previously, he completed his Ph.D. in the Laboratory of Perception and Action at UC Berkeley. Please join me in welcoming Santani Teng.
Thank you. Can everyone hear me okay? Great. Well, pretty soon you can also see. So I just want to say I'm standing here for 3 reasons. One is, of course, I want to thank Natalie and the organizers for putting this on and giving me a chance to talk about myself yet again. And second, why am I here at Smith-Kettlewell? This is really Val's fault, right? She was, we were colleagues in grad school at Berkeley, and when she came here, when I started talking to her about working here, she was relentless in extolling the virtues of Smith-Kettlewell as a place to, really forge your own ideas in terms of anything from basic to applied research that might affect or help or just inform the world of blind and visually impaired people. And third, basically since grad school, I've had a long interest in just the basic neuroscience of cross-modal plasticity, what happens in sensory loss. And I was, raised in a vision lab, and so I've spent a while trying to overcome my visual chauvinism. Okay, so, so bear with me on that. We're all on a journey here, right? Very quickly, there is a world of about 20 years of cross-modal plasticity work showing that visual cortical areas, and I'm sort of pointing to these early visual areas in blind folks, especially congenitally blind or early blind, become responsive to non-visual tasks. And a canonical one that has been published many times is Braille reading. And one of the things that we know pretty well is that this happens. Some of the things we don't know very well is what kind of a network does this form, right? Brain blobs don't just sort of appear by themselves. They're doing something, and they're doing something in an order. So for example, we have the V1, and we have an area called the visual word form area that is responsible for reading words in sighted people. They form part of a reading network. This is, don't worry too much about this data here. This is just one of my favorite figures that shows if you apply a little magnetic pulse to the brain of a Braille reader right after you present a Braille character, about 20 milliseconds in the touch cortex, the somatosensory cortex, you impair their ability to both detect and read the letter. If you wait an extra 60 milliseconds and apply it to the visual cortex, they can feel the letter just fine. That's this flat blue trace here. But they have trouble identifying it. So there are detection and identification processes that we can sort of dissociate here. Of course, what it means not to be able to read a letter is not entirely clear from this, right? Is the dot information at a low level sort of interfered with, or is the lexical information or the reading information at a high level sort of obscured? And of course, there's the question of what, these sort of activations don't tell us what is actually happening there. And this is just a little slide I took from a recent study where visual cortex in early blind people was responsive not to low-level sensory information, but to high-level phonology and grammar, language information. And so this is kind of a semi-raging debate in the community. What is actually happening in these areas, right? Are they all doing sort of the same task, or are they really repurposed in sensory loss? So when I talk about dynamics, I'm not just talking about time, I'm also talking about information. How does the information evolve? So the way we tried to get at this is by having some folks look at letters while being recorded with MEG. MEG is magnetoencephalography. It is a much more expensive version of EEG. And what you get is a little bit of extra signal from that. But basically what we're able to do is measure the brain response at a millisecond scale. Okay. So how do we present these letters? This is just a little something I want to say, stop for a second on. These are refreshable Braille displays. These are single individual letters that we're presenting each time. So we're not able to present whole words, not for technical reasons, but for experimental reasons. We want to be able to control the onset of the stimulus, right? Vision people are spoiled. The retina gets all its information at once in a picture. Maybe not the processing, but the acquisition of a Braille word is sequential in some sense. It takes time. And so if we're measuring the time of the Braille response, of the brain response, we don't want to have that confounded with the actual sensory information. So the subjects were presented one of a series of letters to a stationary finger. It was just sitting there. The letters popped up. So it's not a very natural way of reading, but they were able to read the letters quite easily. We tested them before, and they did a task that I'll mention in a second. So it's essentially a slideshow, right, of Braille letters. We had a group of sighted subjects do the same thing with visual letters. So everyone was reading in their home modality, right, and they were just presented with, this little icon shows a capital T. They were all lowercase letters to equate. And so they did, other than the fact that they were different populations using different modalities, they did the exact same task in the scanner. They were just presented a letter about once every second or so, and in each case the letter was presented for half a second. And then every once in a while, the sighted subjects would see an oddball. They would see a non-letter and they would just press a button. This is essentially to keep them awake, right? To make sure they're reading the task. We throw those trials away, so all you see is the passive reading. The reason is that if you push a button, the muscle artifact sort of blows up the brain signal, and the signal gets lost. Unfortunately, these 6 dots of Braille cells don't have nonsense combinations. Every possible pin combination means something, and so we just asked them to look out for a particular pair of letters that just in this case it was an E and an O, and so so they were reading the letters, and every time an E or O came up, we flagged that trial and threw it away. So they did this for about an hour before they lost patience, and when I was trying to prime you guys a little bit earlier about fancy computational methods. Also, the fact that I did not invent these or develop them, but what this is for people who are interested, basically we do a pairwise linear classification analysis on the MEG patterns at every time point. What that means, okay, don't worry about that or ask me about it later if you want. What that means is that at every time point, that means at every millisecond, starting from, before each letter comes on and all the way out to about a second later, we take the brain response as measured by 300 sensors on the little helmet that people are wearing. And we measure the difference, we compute the difference between every pair of letters, right? So not just what happens when you have an A, but what happens when you have an A versus a B versus a C. In our case, we have a subset of the alphabet, so it's actually B, C, D, L, M, N, and W, X, Y, Z. So sort of a sampling across the alphabet. Okay, what that gets us is a matrix, and this looks fancier than it is. This is just a color code showing that between every pair of letters we get a certain representational distance, and that just means how well can we tell those signals apart. Was that? Oh boy. Okay. This is so what we do is collapse that into just a graph here. Okay. I'm going to end with a movie, and then you can forgive me my time here. Okay. So what we have is in purple. This is the sighted. We have a big onset, quick, as we expect. By about 60 milliseconds, the brain is responding. What we haven't seen before here, and this is one of our new findings, is that this is not the raw brain response, but how well the letters are distinguishable in the brain response, right? That starting at about 100 milliseconds, you have this long, sort of lower level, but sustained response. And where does this happen? I'll just show you this here. This is the same analysis, but just done over the head, okay? Here is our little person facing up. We're looking at the top. And if you just watch this movie, the letter comes on now. And by about 100 milliseconds, you can see the visual cortex active here and the right somatosensory cortex. So where the information comes from the finger. And by about 300 or so, the sighted brain is done. Notice this very ill-behaved signal in the Braille reader. It lingers for a long time. So there's some difference in the dynamics here, right? It lasts all the way to the end of the trial in some way that is hard to characterize, that we're still working on. I want to show you one plot here. This is, before I get played off the stage here. This is a correlation between the time scale on blind braille readers and visual sighted readers. And, oh, I accidentally hid the time scale. This is about 150 milliseconds in the sighted, and a little swath from 400 to 800 milliseconds in the blind. In that patch, in those different time regimes for the two populations who have never had any experience with the other group's task, you get a little pattern of correlated activity. Those brains are doing something similar there. And so we're able to get some sense of the dynamics of the signal. And I'm going to skip this last bit, but what I want to say is just that we can now read out the spatiotemporal dynamics of letter processing, and that with that, we don't just see when, what the brain response does in a raw sense, we see what information it carries, and that it carries the same information at a certain point between these two unrelated populations, as in they were not the same subjects. And that you can sort of read out what the information there is, right? I want to just, little mea culpa, this is a small subset of what Braille reading actually is, right? The subjects weren't moving, it was single letters and so on, but it's a first step. And of course, doing it with motion and everything is much harder. And finally, just these methods give us sort of a new way of looking at how this information is represented. And there's my 8 + 2 minutes. All right, thank you. Oh, yeah. Thank you to all my mentors here, by the way. I don't acknowledge them publicly very often, but Josh, James, and John are my official mentors here. And then these folks at MIT showed me how to do all that stuff that I just showed you. So thank you, and if you have questions, I'm sure you can hunt me down later. Yeah.
Okay, great. Thank you so much, Santani.